Behavioral Neuroscience, Chapter 9, Segment 2. The vestibulocochlear nerve is otherwise known as cranial nerve number 8, which runs from the cochlea to the brainstem auditory nuclei. It is made up of about 30,000 to 50,000 auditory nerve fibers. Each auditory neuron responds to a very precise frequency at its threshold, but for more intense stimuli, the neuron responds to a broader range of frequencies. A tuning curve is a graph of the responses of a single auditory nerve fiber, or neuron, to sounds that vary in frequency and intensity. The brain analyzes signals from thousands of fibers to calculate the intensity and frequency of each sound. Input from the auditory nerve is distributed to both sides of the brain. Each auditory nerve fiber is divided into two main branches as it enters the brainstem. Each branch then goes to a group of neurons in the dorsal cochlear nucleus or a group in the ventral cochlear nucleus. Cochlear nuclei are brainstem nuclei that receive input from auditory hair cells and send output to the superior olivary complex. Superior olivary nuclei are brainstem nuclei that receive input from both right and left cochlear nuclei and provide the first binaural analysis of auditory information. Other paths converge on the primary auditory centers of the midbrain, the inferior colliculi, paired grade matter structures on the dorsal midbrain that receive auditory information. Outputs of the inferior colliculi go to the medial geniculate nuclei in the thalamus that receive input from the inferior colliculi and send output to the auditory cortex. At the medial geniculate nucleus and auditory cortex, neurons are not only excited by certain frequencies, but also inhibited by neighboring ones. This allows us to discriminate very small frequency differences. Tonotopic organization is a major organizational feature in the auditory system in which neurons are arranged as an orderly map of stimulus frequency, with cells responsive to high frequencies located at a distance from those responsive to low frequencies. The most active neurons take up the most 2dG. This reveals which cells were most active when the stimulus frequency was presented. The primary auditory cortex, or A1, is the region of superior temporal cortex in which auditory processing occurs. Studies show that the auditory cortex integrates other non-auditory information with sounds. Speech sounds primarily activate other areas. The minimal discriminable frequency difference is the smallest change in frequency that can be detected reliably between two tones, usually about 2 Hz for sounds up to 2000 Hz. Larger differences are required for higher frequencies. Place coding is the encoding of sound frequency as a function of the location on the basilar membrane that is most stimulated by that sound. Treble is an aspect of pitch corresponding to the subjective experience of high-frequency sounds. It activates receptors near the base of the cochlea. Bass is an aspect of pitch corresponding to the subjective experience of low-frequency sounds. It activates receptors near the apex of the cochlea. The cochlea maps simultaneous peaks of vibration at different places along the basilar membrane. Temporal coding is the encoding of sound frequency in terms of the number of action potentials per second produced by an auditory nerve. The firing of the action potential is phase-locked to the stimulus, meaning it occurs at a particular point in each sound cycle and thus directly reflects the number of cycles per second. Generally requires an averaging across several cells. Infrasound is very low frequency sound, in general, below the threshold for human hearing at about 20 Hz. Ultrasound is high frequency sound, in general, above the threshold for human hearing at about 20,000 Hz. Binaural is referring to two ears. Intensity difference is perceived difference in loudness between the two ears, which can be used to localize a sound source. A sound shadow is preventing sounds located on one side, called off-axis sounds, from reaching both ears with equal loudness. The shadow effect is most pronounced for high-frequency sounds. Latency differences are differences between the two ears and the time of arrival of a sound, which can be employed by the nervous system to localize sound sources. Onset disparity is the difference between the two ears in hearing the beginning of a sound. Ongoing phase disparity is the continuous mismatch between the two ears in the arrival of all the peaks and troughs that make up the sound wave. The duplex theory is a theory that we localize sound by combining information about intensity differences and latency differences between the two ears. Coincidence detector is a device that senses the co-occurrence of two events. This is part of the Jeffress model of sound localization. The LSO processes intensity differences, and the MSO processes latency differences. Sound location is encoded by the relative activity of the entire left MSO compared with the entire right MSO. Spectral filtering is the alteration of the amplitude of some, but not all, frequencies in a sound. Changes are produced by the outer ear. The auditory cortex evolved to process the sounds of everyday life. Evidence suggests different pathways to process what versus where. The Haskell's gyrus, where music is first processed, is more than twice as large in the professional musicians as in non-musicians. Amusia is a disorder characterized by the inability to discern tunes accurately. Diffusion tensor imaging, or DTI, reveals that tone-deaf people have fewer connections between frontal cortex and the temporary auditory cortex. Hearing loss is the decreased sensitivity to sound in varying degrees. Deafness is hearing loss so profound that speech perception is lost. Three main causes of hearing loss and deafness are a failure of vibrations to reach the cochlea, problems with the cochlear conversion of sound waves into action potentials, and dysfunction of auditory processing systems in the brain. Conduction deafness is a hearing impairment that is associated with pathology of the external ear or middle ear cavities. Sensory neural deafness is a hearing impairment that originates from cochlear or auditory nerve lesions. This is usually permanent. Mutations in the gene GJB2 may be responsible for as much as 50% of congenital or early onset hearing loss. Ototoxic is toxic to the ears, especially the middle or inner ear. Tinnitus is a sensation of noises or ringing in the ears. Central deafness is a hearing impairment that is related to lesions in auditory pathways or centers, including sites in the brainstem, thalamus, or cortex. Word deafness is the specific inability to hear words, although other sounds can be detected. Cortical deafness is a hearing impairment that is caused by a fault or defect in the cortex.